Welcome to Science on Tap Minocqua. Science on Tap is an example of the Wisconsin idea, an idea founded in 1905. And it's the idea that the boundaries of the university are the boundaries of the state. And this is set up to be a discussion, not a lecture. So we'll engage the audience quickly as we go. And I would like to thank our partners for Science on Tap Minocqua. That is the Minocqua Public Library, the Lakeland Badger Chapter of the Wisconsin Alumni Association, UW Trout Lake Station, UW Kemp Natural Resources Station, and our host, the Minocqua Brewing Company. There's 4 ways to watch right here, live at the brewery. You can also live stream it from your computer. You can watch it at the library live streamed, or you can watch it in the future from our website, scienceontapminocqua.org. Just so you all are aware, our next event is on March 2nd, and it's Fishes of Wisconsin. And that will be done by a DNR researcher named John Lyons. Okay, tonight we have Dr. Tricia McMahon, and we usually think of folks with a PhD to be pretty smart. And Tricia had a bachelor's degree, a master's degree, and a PhD in engineering before realizing that the really exciting work is in microbiology. And lakes, yes. So a little bit about Tricia. She grew up in Urbana, Illinois, where her father is a professor in electrical engineering at the University of Illinois, Champaign-Urbana. And before switching to natural systems, she studied wastewater treatment, and she still has some projects in that field. And most definitely, she looks at the natural world differently than your average lake biologist. She'll be talking about microbes in lakes, and how even though they're really small, they do really important things. These microbial communities are unimaginably diverse, and they are incredibly important to water quality and nutrient cycling. They may be invisible, but they are essential. So we have our Tricia trivia question. So Tricia decided to become a professor at an early age because, A, she saw how dedicated her dad was to his engineering career, so he must have a way cool job. B, she aspired to an extravagant lifestyle and knew professors were about the best paid profession anywhere. Or C, she knew how highly scientists are regarded in Wisconsin these days. A, yes, A. Following in Dad's footsteps. Okay, so without further ado, Dr. Tricia McMahon.
Awesome, thank you. Thanks for that great introduction. Well, first of all, I really don't like lecturing. So part of what I do as a professor in Madison is to try to challenge the undergraduates to wake up and not just listen to me drone on. So I'm going to start out by asking you a question. How many species do you think live in Lake Minocqua? Give me an estimate. 1 million, that's a good start. Do I hear anyone think lower, anyone think higher? 685 species, excellent. Other guesses? 2,000? Okay, so on the lower end, we're getting a few hundreds. On the upper end, we're thinking 1 million. So there's actually, in the world, or the estimates at a global scale, are that there are 1 million different species of insects, okay? In Minocqua, actually, at Lake Minocqua, I actually have no idea how many insects there are, but, you know, maybe there's a few hundred, or maybe 1,000, or a few thousand species of insects. Estimates are that there are about, in any particular lake, about 100,000 species of bacteria. And so the diversity of microorganisms, and when I say microbes, I tend to be talking about bacteria, and we can talk later about whether that definition really holds or not, but when I think of microbes in lakes, I'm thinking about 100,000 different species or so. Globally, estimates are about 1 billion different species, so contrast that with insects. People get all excited about how diverse insects are, and I say, you know, don't talk to me about insects. People say, I found a new species of frog in Panama, you know, cover of Nature and Science magazine, it's on NPR, and I'm like, I can go out and get a teaspoonful of soil out of my garden and find 10,000 new species of bacteria in there, so don't talk to me about biodiversity. So I am sort of the resident speaker for the microbes at the Center for Limnology at UW-Madison. You know, you hear about the Lorax speaking for the trees. I speak for the microbes, and I try to advocate that they are the coolest, most information-rich and biodiversity-rich group of organisms on the planet, and everybody should be studying them. So how many cells do you think, how many bacterial cells do you think are in Lake Minocqua? Billions? How many billions? 10. Try 10,000 billion. So number of stars in the Milky Way, I looked this up before I came up here and trying to keep the numbers in my head. Number of stars in the galaxy, in the Milky Way, are 10 to the 11. So what's that? That's 100 billion, right? So 10 to the 16 cells in Lake Minocqua. So that's 10,000 billion, right? I think if I got the numbers right. Right, so numbers, biodiversity dwarf everything else. Now, the problem is that we can't look at a glass full of water from Lake Minocqua and see those microorganisms and count their legs or see what color fur they have. And so it becomes really difficult to wrap our brains around, you know, what kind of species are there. We certainly have even more trouble figuring out what they do in the lake. So when I talk about, you know, 10,000 billion different cells of bacteria in Lake Minocqua, what do you think about? Are you worried about going swimming if you know there's that many? Upset stomach, why? What makes us upset stomachs? Something that's not happy in your gut, so probably a microbe that doesn't really belong there. But like what normally, like why would you close a beach? What do you normally hear about when you hear beach closings? E. coli, right? E. coli is the bacteria everybody recognizes. E. coli and other organisms that would disrupt your gut are probably 0.0000001% of those many, many bacteria in Lake Minocqua, if there's really any that you could detect at all. I mean, certainly if a goose comes along and poops in the lake, then there's probably some E. coli. But, you know, relative to the organisms that are naturally there, that are performing a lot of really important ecosystem services for the lake and for us, they're vastly small numbers, right? So the beneficial bacteria that live in the lakes are vastly outnumbering those nasty ones that you probably still don't want to ingest. So trying to figure out what those many, many organisms are doing when you can't see them, we often can't grow them in captivity. They're even harder to reproduce in the lab than pandas, right? So they're really hard to get to grow in a cage. Microbiologists that normally study organisms that will grow in a flask or on a petri dish are usually studying what we call weeds, right? So in microbiology, there's organisms that will grow for you in the laboratory, but at least 0.1% of them, or 99.9% of them, will not grow in the laboratory. So we have this big challenge of not being able to see them, not being able to easily categorize them into groups, or to be able to see what they eat or what they're doing. Yet we know that they're providing these really important functions because we can measure some of those parameters, those functions, by doing certain kinds of experiments with lake water. But trying to assign those things that we know that they're doing to different species is really, really difficult. And so one thing that we've started doing in the past 5 to 10 years, sort of we as a community of researchers who study microorganisms that live in all kinds of different habitats, is just sequencing their DNA. So 13 years ago, the Human Genome Project finished. A lot of the technology that was developed to sequence human genomes is now driving the science forward in microbiology. So we can go and take a glass of water from Lake Minocqua and we can sequence all of the DNA in it. We can look at every last little piece of DNA in it. And we can tell when we do that sort of what kinds of organisms we've seen before, what kinds we've never seen before, and we can kind of start to group them into sets of organisms that we think are performing different kinds of functions. So what do you think would happen if we suddenly took all of the bacteria out of Lake Minocqua? What would happen? Yeah, probably it would be very clear. You're right. Well, for a little while, then what would happen? No plants or fish, right, because they sustain a lot of the rest of the food web. But eventually, so 5 years, 10 years down the road, what would happen? Maybe 1 year, what would happen to Lake Minocqua? Well, dead. What does dead mean? There's no other organisms? Maybe. Well, I already told you, didn't I, over dinner? Well, eventually, I mean, assuming that the other life forms could exist without the microbes, but when they die, they would just fall to the bottom and start to pile up, right? So eventually, the lake would fill up with other dead things, dead fish, dead plants, and whatever. So the microorganisms play this critical role of recycling. So maybe you've heard about them as being decomposers or being, other kinds of organisms at the bottom of the food web that kind of eat all the dead stuff, right? So if they go away, you don't get recycling of the nutrients, you don't get recycling of the carbon, you get a lot of material that would just accumulate in the lake, and eventually it would fill in and become land, right? So the microbes play that really critical role of recycling. Of course, that can sometimes cause problems because if they recycle something like phosphorus or nitrogen back into the water, that becomes available for algae, which can fuel algae blooms. So they are providing this critical role, but they also are helping the ecosystem to kind of keep spinning, which can sometimes have these consequences that we don't necessarily value as being a good thing for the lake. Same goes for something like anoxia. So in the summertime, a lake that's deep enough will often stratify, where it's warm at the surface and cold at the bottom, and it's a very stable system because of the density of water. That's a very natural process. Microorganisms at the bottom of the lake will start to use the oxygen and eventually deplete it. You know, if the lake is productive enough, if there's enough algae growth happening in the lake, you will get depletion of the oxygen at the bottom. And that is absolutely the fault of the microbes, although you can argue about whether or not that's really their fault because they're just doing what they're supposed to do, which is to break down the dead stuff. They happen to breathe oxygen the same way that we do. Well, not exactly the same way, but same principle. The cool thing about the microbes at the bottom of the lake is that they can breathe other stuff too. So when the oxygen is gone, they will actually breathe nitrate, which is a nitrogen compound, and recycle that. They will also breathe sulfate, which is a sulfur compound, and recycle that. They'll breathe iron and turn that into a different form of iron. Eventually, they will breathe dead things, right? So they basically are eating dead things, but then also breathing dead things. And sometimes when they do that, they make methane, which is maybe not such a good thing because that's a very potent greenhouse gas. But luckily, there's some organisms at the top layer that eat the methane before it gets out of the lake. So you could imagine taking away any of those pieces and that whole system falling apart. So one of the things that we're finding by looking at the DNA of these organisms is that they actually function more like a superorganism. So you can track each individual species as a unit, but it itself can't do everything that it needs to do in order to survive or to reproduce and be a living organism. It depends on its neighbor to provide some essential function. So probably the best example is making a vitamin, right? And we all need other organisms to provide certain vitamins for us, right? Either the organisms in our guts or it's in our food that's produced by an animal. Same goes for the communities of bacteria that live in Lake Minocqua. They are depending on each other to provide different intermediates that they need for their growth. So if you try to take any one of them and grow them in a flask in the laboratory, it doesn't work because they need their friends around. And so it's through all this syntrophic interaction that's happening with these communities that you get the breakdown of the dead fish, the breakdown of the dead plants, dead algae, recycling of the nitrogen and the phosphorus into something that the food web can use and send back up to the fish, right? So this is one reason why they're so hard to study, is because we can't separate them and we can't do the kinds of experiments that most microbiologists or most biologists would do on an organism in order to figure out how they work. So some of the biggest research frontiers now are trying to figure out how this superorganism collection of individual organisms function, how they change through time, how they respond to conditions that we know happen on a seasonal time scale. So when the water warms up, when it rains and there's an influx of phosphorus or some form of nitrogen, how do they respond? If you suddenly have a season where the lakes don't freeze anymore, how are the organisms going to respond to that? And because we don't understand all the inner workings of that black box of that community, we're working really hard to try to unpack and tease out what's happening there. And that's really the research frontier of studying any kind of microorganisms in any ecosystem, but definitely in lakes. And so we're doing that in Lake Mendota, down in Madison, of course, but we also have several projects up here tied to Trout Lake Station, trying to figure out how the bog lakes function in particular because they're sort of carbon emission hotspots, and microorganisms are very responsible for that. But also trying to understand how oligotrophic lakes, so the really blue, beautiful ones, how microorganisms might to some extent control the transition from being beautiful and clear and blue to being green and scummy and yucky, and nobody wants that. So trying to figure out how the microbes play a role in mediating that. So I promised that I wouldn't go blah, 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 lecture, lecture, lecture. So I will see if we have any questions now that I've given you some food for thought.
Hi. Sometimes when I'm in the reaches, the far corners of a lake where it's really shallow, it almost seems like a part of the bottom of the lake that is about this thick has sloughed off of the bottom and is floating around. What's in that?
Ooh, that's a great question. Probably the best term that I would use is a biofilm. So it's probably a mixture of bacteria and, periphytins, so algae that grow on surfaces rather than the kind that float around normally free living. Maybe even some microscopic animals and, invertebrates and things like that. But the bacteria are the ones that make that structure, that sort of gooey, oozy, fluffy stuff that holds it all together. And they're doing it partly because life attached to a surface is actually usually better than life floating around alone. And that's because their neighbor might be making something that they need, or maybe the algae next to it died and is now breaking open and releasing a bunch of yummy food for them. And so it's actually, most microorganisms prefer to live aggregated with other organisms rather than floating around on their own. And why it comes detached all of a sudden, that could just be a physical, like a wave came by or somebody's boat, or it could be lots of different reasons why it would become detached. But it is a lot of bacteria holding it together, gluing it together. Is it usually a weird color? Is it brown? Yeah. Brownish, greenish, yeah.
Okay, I have a question. You talked about stuffing it in my mouth.
Stuff it up.
I got to, but remember, it's got to be out like that.
Okay. How does climate change, which means extra rain maybe, whatever, how does that affect the lake and the phosphorus load and other things that could happen? And how do you think the bacteria and the microorganisms can handle that?
Yeah, that's an excellent question and something that we're really interested in researching. And it probably, as usual, as a scientist, I will say it depends, right? Where you are, it depends on what kind of lake you're talking about. I think about climate change and lakes mostly in Madison. I mean, we can talk a bit about what would happen up here in northern Wisconsin, but certainly in Madison, the phosphorus inputs to Lake Mendota and the other Yahara lakes very much dictate what the algae bloom situation will be in the summer. And the springtime rains and how much phosphorus comes from the farmland, and that might be sort of stored in the sediment of the rivers going into Lake Mendota, are really what determine the cyanobacterial blooms, the algae blooms that we see in the summertime. And so if you get a big rain event at the wrong time, which is exactly what some of the predictions and trends related to climate change are telling us will happen, that can be the biggest source of phosphorus into the lake all year. If you get it at that moment in the spring when the ground is still frozen and manure has been spread and the water and the rain just washes it into the river and then into the lake. How microbes respond to that, I think probably one thing that we have figured out that we're pretty sure about is that microbes in lakes are very resilient. They will keep doing their thing. They don't care how much rain you get or how much phosphorus you get. And they will do their job of breaking it down and releasing it and making it available for the algae and the cyanobacteria. They're not going to stop doing that as a result of something that would happen with climate change. I think figuring out how fast they do it, figuring out whether or not they do it before the phosphorus has a chance to sink down to the bottom of the lake, for example, are questions that we have to ask in collaboration with people that study the physics of how the water moves, and through thinking about how that chemistry of the phosphorus interacting with the water, we have to think about all of those things as well. So one reason why I love microbiology so much is that it's something that you can't do just thinking about biology. We have to always be thinking about the chemistry of the system, about the physics of the system, and in my case, I think about engineering, so I think about sort of how it functions as an engineering-like system. But I'd say that we don't know how the microbes are going to respond to those climate change drivers. But they are surprisingly resilient. We did a neat experiment here up at Trout Lake in 2008 where who here has heard about the Crystal Lake Mixing Project? Okay, right, so similar to that. In fact, the devices that were used for the Crystal Lake Mixing Project actually were prototyped in a project that we did on a much smaller lake where we artificially mixed it in the summertime to see what would happen to the microbes. And what we found was that they were disturbed by the mixing, so they changed in terms of which species were present. They changed in a way that they normally would not at that time of the year, but that change only lasted for about a week, and they went right back to where they were before. So that, in a sense, was good news because we're like, okay, well, the microbes can deal with it. So I think the concern about microbes and climate change is probably more about what the feedbacks are going to be, because microbes do a lot of they release a lot of carbon dioxide when they're doing their job of breaking things down, and so if that gets accelerated because of something else is happening in the ecosystem, that could cause feedback with accumulation of CO2 in the atmosphere. Things like that.
So you talked about the DNA. How closely related are each microorganism in the water? And then, we always hear that we've got banana DNA in us, and so how close related are they to us? And then I really want to know, like, to each other, like, how much are you differentiating between one and the other, and how do you know that that's that one and that's that one?
Oh my gosh, I could talk for 3 hours about this. So the way that we have studied them sort of up until we could do all this whole genome sequencing was that we look at a single gene that they carry, and that gene they share with every other cellular life form on the planet. So we have it, bacteria have it, fungi have it, plants have it, everything has it. And that piece of DNA, that single gene, has parts of it that are almost completely identical in all life forms. But there's probably only about 20 bases of DNA where it's completely identical. But you can line it up and look and see that it looks just like ours. So that, goes back to the last common ancestor of all cellular life. But at the level of 2 bacterial species that you compare those 2 genes, or those genes from each of those 2 bacterial species, they would probably be about 97% identical, which is about the level of identity that we share with other primates, right? So, and even there, we're not talking 2 different species. When you're talking about a human and another primate, it's like a different family, right? So the way that we categorize bacteria into species is very conservative. Probably if you were going to apply the same standards of how similar do they have to be in order to belong to the same species that we do with eukaryotes, so multicellular organisms like us, then there would probably be, 1 million times more species of bacteria than we've even estimated. So we're very conservative in how we call a species a group. Now at the genome level, so if you're comparing whole genomes, which they evolve at a different rate than this specific gene, 2 bacterial species might be about 85% identical across their entire DNA sequence, their entire genome sequence. And I actually am not sure what humans and primates would be. I don't have that number in my head, but certainly we're looking at that level of identity between species. So if you're talking about different, if you go back to your freshman biology and you're thinking about there's species, genus, test me now, right? Family, order, class, right? If you start going up that level of Linnaean taxonomy, then you're starting to talk about more like 70% identity at the genus level and maybe 60% identity at the family level, and to the point where you can't really line them up anymore because they're too different across their whole genome, whole piece of DNA. And then there's lots of interesting things that you can do to look at how many genes they share. Remember I said that as a community they're kind of like a superorganism. So you might have a species over here that has 1,000 genes, and this species over here has 1,000 genes. But and the overlap between them might only be 100 genes, right? So each of them is bringing sort of a unique set of 900 genes to the table. But the 100 are what overlap. So as microbiologists, we spend a lot of time looking at these diagrams showing what they share, what they don't share, what can this one eat but this one can't eat because it doesn't have that gene, and trying to piece all of that together.
Yeah, in the Clean Water Act in the 70s, it was either 72 or 78 or somewhere in there, the government wanted Superfund cleanups and kind of went after corporations, paper mills in Wisconsin here and other things. At the same time, that bill exempted all agriculture and ranching, pretty much all of it. Maybe they didn't all of it, but for all intent and purpose, they exempted that. And you were speaking about Lake Mendota. And I was told once not too long ago that the major pollutant in Lake Mendota was the O'Hara River watershed, which is 20 to 22 miles long and 20 miles wide or something like that. And is basically stuff coming off farm fields and everything else. And I lived in Madison in the 70s and early 80s when Lake Mendota was relatively clean. And now it's gotten pretty grungy. Another area is Tainter Lake, Lake Menominee on the Red Cedar River in Dunn County, which in the summertime in August you can mow and walk on.
It's one of the worst in Wisconsin,
Yes.
yep.
And that again is from the Red Cedar watershed. There is very, very, in comparison, little industry. And I know that you have to go at each one of the farmers gently, delicately, and with a lot of knowledge to help them see the light about cleaning that stuff up. Do you think there's going to be another bill sometime, somewhere down the road with clean energy, a clean water bill that's going to address agriculture?
Yeah, it really comes down to non-point versus point source pollution and how hard it is to regulate and hold someone accountable for the non-point sources, right? So having enough surveillance to know that a particular farm is discharging, X amount of phosphorus compared to this other farm and where it goes and how it gets there is just, certainly when the Clean Water Act was written there, the technology did not exist to have that level of surveillance, and so you couldn't have the enforcement, and so it just was not feasible. I'd say we're getting closer, if you want to get really creepy about it, you could imagine drones like flying over and like taking pictures and trying to map where all this happens. There's certainly also in situ sensors, so people are building autonomous devices that you could set up in the stream or something that would at least detect, if phosphorus was high or low upstream of a particular place where a farm runoff would be coming in. But even that would be a big infrastructure investment to do that level of surveillance. So I think that we're still pretty far from being able to enforce any regulations that would relate to non-point source pollution. So I think that's the main barrier to having that kind of legislation and actually have it be meaningful. Has anybody come to talk about what's being called the Adaptive Management Project in Madison or in the Madison area lakes? So this is kind of on the edge of what I do, certainly not in my research, but I did serve on the Dane County Lakes and Watershed Commission for a few years, so I know a bit about it. Basically, there's an initiative going on in Dane County where, so let me back up a step. The Fox River Basin had a total maximum daily load rule be administered by the EPA. So what that means is that the Fox River Basin had a certain amount of pollution in their waterways, and so the EPA said to the DNR, you have to come up with a plan to clean this up. And so the DNR had to stipulate how much phosphorus and how much total suspended solids, so like the particles that would enter a water body, how much of that could be entering from various point sources, so wastewater treatment plants, industries, stormwater outfalls that are managed by cities and other municipalities. So each entity like that in the Fox River Basin had to meet those new regulations set by the DNR. And this was obviously a big onerous thing for some of them, right? Really expensive to have to meet these new limits. And in particular, the wastewater treatment plant in Madison, who I have to disclose I do work with a bit, had they crunched the numbers on how much money they would have to spend to upgrade the wastewater treatment plant to meet those new limits, and it was, hundreds of millions of dollars. And they're like, this doesn't make any sense because that wastewater treatment plant actually discharges south of the Yahar Lakes. So they could spend hundreds of millions of dollars upgrading the wastewater treatment plant. It would have no impact on the Yahar Lakes, right? They said, this is a waste of money. Why would we do this? So they and others went to the DNR and said, can we have permission to come up with a more creative solution to this TMDL problem, this total maximum daily load problem of how much phosphorus and particles enter our waterways? And the DNR actually gave them the flexibility to try something pretty new. And this is something that's being watched all over the country as a model for how to deal with water quality and in point source and non-point source pollution. So what they did was the engineers at the wastewater treatment plant went around and talked to other wastewater treatment plant operators, mostly in Dane County, to the cities, City of Madison being one of the biggest dischargers of stormwater to the Yahara River, or Yahara Lakes, of course, and talked to all these entities that were going to be affected by this new rule and said, let's band together and use our money to fund projects in the agricultural part of the watershed that's north of the lakes, to come up with best management practices. Actually, most best management practices for agriculture are well understood. It's more about implementing them. So let's use our money that we would have spent upgrading our wastewater treatment plants and spend it on fixing the real problem, which is, a slope being too steep coming off of a farm field so that it runs straight into, a tributary to the Yahara River. And the DNR said, you can do that, that's fine. You have to prove that what these interventions that you're doing, these changes that you're making, that you're paying for, actually have an effect, that they actually are reducing the load of phosphorus and particles going into the lake. And they said, great, we can do that. And so they built a pilot scale system to try to test it out. And the preliminary results for that were very promising. And so now they're moving into the next phase where they're expanding it and spending more money to try to do this. And it's created all these really interesting things to think about and study because, first of all, it's a model piece of I wouldn't say regulation, but enforcement of regulations, where you're having the people who would be normally regulated sort of spend their efforts on someone who's not being regulated, the farmers, to try to fix their problem so that they don't have to waste their money fixing a non-problem, right? So that in itself is a really creative and innovative way to address that challenge. But it's created all kinds of weird issues that come up sort of socially and politically because, for example, Fitchburg, right, south of Madison, Fitchburg, their municipality is paying money to fix something in north of Lake Mendota, and so people are seeing their tax money go away from Fitchburg to do something that they may never see the benefits of because they're not even on the lake, so why should they care, right? And so there's some objection by some people to this kind of model because it's sort of taking tax money and taking it away from the entity that's collecting the taxes. But the alternative would be that Fitchburg has to pay to upgrade their stormwater system or contribute to upgrading the wastewater treatment system. So there is an offset of expense. But I think a lot of people are watching it. As usual, Wisconsin's kind of blazing a trail in terms of environmental regulations and trying to do it a different way, a more effective way. So all of us who study lakes and water quality are eagerly watching to see if it works. But I think it's kudos to the DNR for letting them do it. It's kudos to the people organizing the wastewater treatment plant operators and the cities to actually going forward and doing it. So we're pretty excited about it. That was a long answer, but I think it's really cool. So like I said, I talk about it for a long time.
So we have tens of thousands of different bacterial species in our lake, most which will not grow in culture. Mm-hmm. So are you defining them by their DNA sequences?
Yes, we are defining it by their DNA sequences and mostly by that gene that I said is shared with all life, right? So again, that's a very conservative estimate.
So what is the Eve gene?
What is the gene?
Yes.
It is called 16S ribosomal RNA. So if you know much about how, so there's this thing called the central dogma in biology where DNA makes RNA makes protein. The thing in a cell that makes protein is called the ribosome, and it's a mixture of proteins and structural RNA. And there's a piece of that, the structural piece of RNA, that is the thing that's in all cellular life. Because all cellular life has ribosomes, and there's so much selective pressure on that gene that it can't change. Because if it changed through evolution, all that machinery would fall apart. And so that's why there's pieces of it that are identical in us and in bacteria. And in fact, there's these microscopic organisms called archaea. Who's heard of archaea? Does anybody remember the 3 domains of life from freshman biology? See, when I took biology, it was, and I'm not that old, right? You're
29.
Yeah, 29. I'm 29. When I took biology, there was 5 kingdoms, right? There's, I don't even remember what they are. Animals, plants, fungi, Monera. What was the 5th one? Fungi. What did I say that? Anyway, you know what I mean. 5 kingdoms. Totally irrelevant now. Biologists categorize life into 3 domains: bacteria, archaea, and eukaryotes. All multicellular life is eukaryotes, so animals, plants, fungi, all the things that used to be in sort of separate kingdoms are now like lumped together in one thing. And archaea, when you look at them under the microscope, look like bacteria, and for a long, long time people thought they were the same as bacteria. But it turns out that archaea, even though they're unicellular and microscopic and look just like bacteria, share more in common with us than bacteria share with them. If that makes sense, right? And that just has to do with the way that diversification has happened and the tree of life branched. So archaea are actually this really fascinating group of organisms that not very many people study. They got famous because you find them in hot springs and weird exotic places like the bottom of the ocean, but we do have them in the bottom of the lake, so they live in the sediment of Lake Minocqua. So there's lots of archaea there. They're the ones that make the methane. And so understanding how they function and when they make methane is actually really important because methane is such a potent greenhouse gas. And so a lot of people study the ones that make methane very intensively.
How do you identify what a microbe can eat and what it breathes?
That's a great question. So traditionally, we would, if we could grow them in the lab, we feed them something and we measure what comes out. And so that's the way you can feed them different things, and if they eat it, you can measure the product. So if you give them sugar, you can see if they make CO2. So you can actually measure how much sugar goes away and how much CO2 gets produced. Now, if you can't grow them in the lab, like I just told you that 99.9% of them can't grow in the lab, it's a lot harder. So we could take a glass of water and put sugar in it and then measure how much CO2 comes out, but then you've just measured what the whole community can do, not what the individual species can do. So some of the techniques that are being developed now are using isotopes of elements. So sometimes they're radioisotopes, so a radioactive form of carbon or a radioactive form of phosphorus or something like that. But then there's also stable isotopes, which are not radioactive, but you can trace, probably the best example is carbon that is, it's called C13. So carbon normally would have a molecular weight of 12, but if it has a molecular weight of 13, you can differentiate it from the normal carbon. And so if you give a particular community of bacteria sugar that has C13 carbon in it, you can track that C13 in individual cells using a certain kind of microscope and see which cell ate it. And so you're doing an experiment where you're having to use this really sophisticated piece of equipment to follow individual molecules of the food through into the cell. And you can do the same kind of thing with what they breathe. But they're very tedious experiments. And I have to say that most of my graduate students would much rather look at a bunch of DNA sequence and sit at their computer rather than try to do these really complicated experiments. So that's what a lot of us are doing these days, is more of the sort of trying to interpret the DNA sequence. If you look at the DNA sequence, you can see a gene that the organism had, and it's probably related, it might be related to something that's another organism has that has been studied in great detail. E. coli, even though it's the one that makes you sick and everybody knows about for beach closings, also is the lab rat of microbiology. And a tremendous amount of research has been done on E. coli. And so we know that E. coli can breathe nitrate, and we know what genes it uses to do that. And so if we see a gene from a bacteria in Lake Minocqua that, is maybe 60% identical to the one in E. coli, then we can start to predict that that's what it's for. You can never be absolutely sure until you do some really sophisticated experiments, but a lot of what we do is making predictions about how what we see in a lake sample is related to something that's already known and what we think it might be able to do as a result of being known to have that function. So we can see the genes that are responsible for eating glucose or for eating, what's another good example of, a lot of bacteria eat acetic acid, which is like what's in vinegar, right? And so you can see the genes that are responsible for that in their genomes and know that they probably eat that.
Okay. A lot of lakes have, and up here, have Eurasian water milfoil. There's a few that have milfoil. They treat it with chemically. They chemically treat it. Now, I understand that those will die, and of course that type of bacteria that will eat decaying plants will be there. Have any studies been done on what kind of effect the chemicals might have on the rest of the bacteria that are there?
I think general studies are often done just to make sure that a chemical doesn't completely inhibit breakdown of dead things, right? So if you put the chemicals in and then nothing happens, it just stays and nothing happens, then you'd be a little bit worried, right? But usually, by the time it's approved to be used in an actual lake, someone has done the experiment to show that it doesn't stop the bacteria from doing that job of breaking it down. A lot of the chemicals that kill plants, though, might also interfere with algae or cyanobacteria, and so there may be some interruptions to the kinds of functions those organisms perform, and it depends on if you categorize those as microbes or not. A lot of people might actually call them microbes, in which case, so yeah, there might be some effect on the phytoplankton, so the organisms that are the green, free-living, floating organisms. It probably depends on what chemical they're using. But again, bacteria are really resilient. It's like people have asked me, well, why can't we just dump a bunch of antibiotics in the lake and kill them all? And I'm like, well, probably not a good idea for a lot of different reasons. But even if you did that, there'd be thousands and thousands of species more that, are there that are naturally resistant or that become resistant or that come from the air and recolonize, right? So keeping the microbes out of the lake is impossible.
Question back there?
Yes.
Whew. Wait, I'm looking.
Back here.
Oh, in the back. Hey,
Okay.
yes, I see you.
Okay.
Lights are bright.
I don't know what happened there, but with all the research that we have come across so far and developed and what have you, how far are we away from actually manipulating the microorganisms to benefit us?
You mean specifically the lake or in general, more general? In the lake. I think we're pretty far. I think there's crude ways that we can manipulate the lake, which would then manipulate the microbes. Best examples are, putting in some kind of aeration system in order to prevent the anoxic conditions at the bottom of the lake. So then you're manipulating them to never get to the point where they're doing sulfate reduction or nitrate reduction and things like that. The breathing these other carbon or other sources of ways to breathe. But I don't think we understand the fundamental interactions of the organisms or how they respond to environmental change enough to be able to manipulate them. As an engineer, I often find that really unsatisfying because I came to engineering because I wanted to solve problems, and I deal with this with my graduate students as well, who come to me as engineers who want to clean up the lake. And we regularly, pull our hair out saying, what are we learning that will actually allow us to do something different with the lake in order to manipulate the microbes and have them do something different? And I think we're still pretty far from that. And as a typical scientist, I'd be like, oh, we need more research money. We need give me a big fat grant and I'll solve that problem. But I mean, I think it's true that there's a lot of basic science that needs to go into it before we can think about how to actually manipulate it. There are some microbiologists who are really excited about a field called synthetic biology, where basically you can build your own life form. You can make a microbe from scratch almost, or you can certainly take an existing microbe and reprogram it to vast extent. But the ethical issues of whether you would release a microbe like that into a lake are, I think that there's so much that we would need to try to understand before we would ever do that. You know, everybody's terrified of, not everybody, a lot of people are maybe unjustifiably terrified of genetically modified organisms in their food. So you would never get permission to release a genetically modified microbe into a lake in order to do something to clean up the water. So I think we're a long way from that. What I get most excited about is figuring out a way to manipulate the ecosystem in a natural way. So you're doing some kind of manipulation physically or with some chemical maybe that's not going to cause that's not toxic to other life forms, but somehow would manipulate the organisms in order to get them to figure out how to do what you need them to do, rather than putting in a non-native organism and taking the risk that something unexpected would happen. But there are people who love that approach and are working on that.
Some of what you said reminds me of just some of the difficulties of science, of trying to isolate the individual behaviors of species and how difficult it is to do that, especially when you're dealing with microbes versus cheetahs or something. And so I'm curious about just the extent to which we understand communities of microbes, because they're the ones, at least if I understand what you're saying, that are providing the ecological services that are already providing these huge benefits for us. But is the science evolving enough to study these communities in a way that it might not actually be able to study individuals?
Yeah, absolutely, and I think that this really is sort of the edge, the frontier of our work right now, we being the larger scientific community. And it's very much being driven by the DNA sequencing technology. But conceptually, I think many of us are trying to approach it from a more holistic sort of system level. And I think this is where, as an engineer, I look at it a little bit differently, because as engineers, we're trained to think about how any one part of a system impacts every other. Microbiologists, their training can be sometimes quite reductionist. They want to get this single organism, study this one gene, and what does that gene do, and let's take it out and see what happens, and let's, getting really in the nitty-gritty reductionist detail. And I think that if we tried to do that with these communities, that it's going to take us, a million years to get anywhere. So you've got to approach it more from a systems biology perspective, and systems biology is its own field in of itself, where it looks at the entirety of an organism and its genome and how its metabolic systems work. But you need to scale that now to a community where you've got many of those systems within systems. But there are people who are coming up with some really fresh ideas on how to tackle that, and so I'm really optimistic that within the span of my career, that we'll figure out what appropriate scale to work at in order to be able to understand enough of it to make predictions about how the system will behave under different new conditions, which is really what we're trying to get at, right? So if the lake temperature goes up by 3 degrees in the summertime because of climate change, like how will the microbes respond to that? So I think we're, within the span of 10 to 20 years, we'll get there. But it's a lot of so thinking it's a lot of big data, a lot of complex connections and interactions that is going to take sort of a new level of training of our graduate students to be able to think about. Because a lot of the graduate students come to me without that training in, so they're having to learn that. And so that itself is kind of a challenge.
I have a two-part question. First is, how long do microbes live? And then the second part is, Do microbial communities change week to week or month to month or decade to decade?
Did someone plant those questions? I did to ask you to, oh, those are such good questions. Okay, so when I started as a professor at Madison, we did not know, I'm gonna take your second set of questions first. How quickly do they change, like from week to week? When I started as a professor, we didn't know that. I'd say now we have a really good handle on the time scales that they change on. So they are very seasonal. In a lake, if you go out in May, the organisms that are there are really different from the organisms that are there in October. And we know that they change on probably like 4 to 7 day time scale. So that means the graduate students have to be out there about once a week taking samples in order to capture that variation. And we know that there are certain times of the year where they change faster, right? Usually when it's warmer, because life happens faster when it's warmer. But we do have a sense of sort of what that turnover rate is in terms of the community changing. Now, how long do they live and why, or what's the mechanism of that change is a little harder to get at. This question of like, if you have a cell and it's doing its thing, it's eating sugar and it's making carbon dioxide and it's just sort of doing its thing, does it ever die, right? I've asked really famous microbiologists this question, and nobody ever really gives me an answer because I don't think anybody ever really, it's really hard to study that. Because certainly in a lake, the problem is that a virus comes along and kills it before it just ages. Like, does a bacterial cell age, I think is sort of the question that I've always had. And I don't think they have a chance to age. I think they get killed by a virus, they get eaten by something else, maybe they get damaged by sunlight, and so maybe you could argue that's aging because they're getting oxidatively damaged, kind of the way our skin gets damaged and skin cells get damaged. But usually it's some kind of mortality that's coming from an external force rather than just, they don't really wear out. They usually die by predation or some chemical stress. But I think that we have a very poor handle on how quickly that happens and is it species specific. We know that viruses that attack bacteria are very species specific. So people who study viruses like to brag about how many viruses there are on the planet and how many species of viruses there are. There are many orders of magnitude even more than the bacteria. And we have even less of a handle on their mechanisms. And the viruses are doing, I know I'm going to go off on a tangent, but they're doing really amazing things with the bacteria where they actually will carry a gene from one bacteria to another and give that new the recipient bacteria the ability to do something that it couldn't do before. And probably the best example are these cyanobacteria that live in the ocean where there's a virus that brings a gene involved with photosynthesis. And the cyanobacteria actually cannot completely do photosynthesis unless they are infected with that virus. And so the virus is not just a predator, it's actually bringing a symbiotic function to the cyanobacteria, which is just sort of like mind-blowing. But so I'm sure things like that are happening in lakes too, but people haven't looked. So.
When we hear about elemental mercury entering the environment and being acted on by bacteria to become an organic form that can bioaccumulate, what's actually happening?
Yes, so that's another example of where maybe they're not providing such a good ecosystem service, but it's they aren't really doing it on purpose, so we shouldn't really blame them. So I always get kind of sad. I actually have a research project studying this, so I'm like, I shouldn't defend them too much, but remember how I said different bacteria will breathe different things. So when the oxygen goes away, when the nitrate goes away, when the iron goes away, and you get down to sulfate. So sulfate is present in most aquatic ecosystems. It's higher in some lakes than others, but there's almost always bacteria around that can breathe sulfate. And in the process of breathing sulfate, they accidentally put a methyl group onto mercury, and they make methyl mercury, so the organic form of mercury. So they do it as kind of this side reaction. And so if you don't have sulfate breathing going on, it's called sulfate reduction. If you don't have sulfate reduction going on, you shouldn't get methylmercury production. And so one way to try to control it would be to prevent all of those other sources for breathing or things that microbes would use for breathing to not become depleted, right? So if you keep oxygen around, you shouldn't get any sulfate reduction, so you shouldn't get methylmercury production. You know, doing that in all the wetlands in northern Wisconsin is not very tractable, but at least there you could imagine an intervention that you would do to manipulate the community so you don't get the production of that toxin. But there's also interesting things breaking that paradigm. I just told you that you have to have sulfate reduction happening in order to get methylmercury. Of course, one of the projects we have going on now that's funded by Wisconsin Sea Grant is to look at, in the Great Lakes, there are pockets of hot spots of methylmercury production in the water column of the Great Lakes where it doesn't appear that these sulfate-reducing bacteria are very active. And so it's sort of challenging that paradigm that these are the organisms that always do it. But we don't know which organisms are doing it. So right now, if you crack open a textbook, it says sulfate-reducing bacteria. But I think there could be other sources that we're not aware of. But that really is, I think, the main dominant source, certainly in lakes in northern Wisconsin. So yeah, one bad thing they do.
Thank you, Doctor. My question is, what would be the relationship between naturally occurring phosphorus, say from a tamarack swamp or whatever, into a seepage lake? What relation would that have to go with oxygen levels under ice during the winter time?
Yeah, so you get a lot of primary production happening in the summertime. So algae and cyanobacteria use that phosphorus to grow. In the process, they're taking carbon dioxide out of the atmosphere and they're putting it into the water. They will sink, they die and they sink, and they end up towards the bottom of the lake. Some of them get broken down in the summer, but some of them just sort of pile up at the bottom. And when the ice forms, the bacteria that are in there just keep going, and they'll slowly break down all those dead algae that grew during the summertime. And they breathe oxygen while they're doing it, and they breathe it till it's gone. And that's when you get anoxia. It's a slower process in the winter because it's cold. It's slower than it is in the summertime. But the balance of how many algae and cyanobacteria, so the phytoplankton, the ones that do the primary production, how much of them grow in the summer and then sink and get decomposed before the ice comes on determines whether or not you get that anoxia happening with the fish kills in the wintertime. Now, there is some evidence that you can actually have algae growing under the ice. So there's enough light penetrating through the ice and the snow on the ice that you can get some of that phosphorus being turned back into live algae, which then will sink and need to be decomposed by the bacteria, which could also then further draw down the oxygen concentration. But on the other hand, the algae make oxygen, so they're actually replenishing the oxygen under the ice to some extent. So I think a lot of it has to do with the timing of when those algae blooms happen in the summer and the fall and when the ice comes on, and just how much total phosphorus went in there to support the growth of the algae. So if you have less phosphorus, you get less algae growing, you get less dead algae, you get more oxygen because there's not as much being used up. So it's that balance that gets upset when you add too much phosphorus. Does that help? Yeah. It doesn't matter if it's natural or if it's human sourced, right? Phosphorus is all the same. So it doesn't really matter where it's coming from. Sorry.
You've been involved with so many projects through the course of your career. Which one has been the most successful or that you're the most proud of?
Depends on how you define success, right? Most proud of. Well, I have two. I don't know if I can decide between them. So I really do have two parts of my research group. One studies lakes and one studies wastewater. And I probably have one from each. Probably, if you really forced me to choose one, there was one that one of my a study that one of my PhD students did. He was my very first PhD student. He's now a professor at UW-Milwaukee, also studying water, more in the Great Lakes, and also aquaculture. He wrote a manuscript, a paper, a journal article, which is the bread and butter of academics, and we wrote it with several other people as well. But it's this huge thing. It looks like it's not quite a textbook, but it's approaching that length. And we wrote it thinking of it as a field guide, right? So if you want to go identify birds, you have a field guide, and you can go and you can see based on the colors of the birds or its beak or whatever, you can look it up and figure out what it is. We didn't have one of those for microbes in any ecosystem. And so we sat down, we said, we want to write a field guide for bacteria in lakes. And so it's this mind-numbingly boring thing to read because it really is just like, this bacteria eats this thing has this DNA sequence, blah, blah, blah. So it's so boring to read. But it is the paper that has come out of my research group that has impacted the most other scientists. So other scientists read it, use it in their work, because if they find a particular microbe in their lake, they look it up in our field guide, and now they know kind of a little bit about what it does. A practical application. I don't do anything practical. Okay, so on the lake side, I'll keep going back to the lake side. I mean, the wastewater side is a little bit easier to talk about because we're talking about lakes, though. I had a graduate student who finished about 3 or 4 years ago who was studying cyanobacteria in Lake Mendota. And the understanding of how cyanobacteria were functioning in Lake Mendota at that time was a certain way. We believed that they weren't doing anything called nitrogen fixation. And he showed that the cyanobacteria were doing nitrogen fixation. And what's more is that the nitrogen fixation was tightly linked to the production of toxins. So cyanobacteria, under certain conditions, will make really potent toxins that will kill you if you drink too much of the water. And he figured out that this starvation of nitrogen is linked to nitrogen fixation, which also triggers the production of these toxins. And this was something that nobody had really put together before. I mean, the pieces were kind of there from other studies, but he brought it all together and showed that it was happening in Lake Mendota. And I think for a lot of other people studying cyanobacteria, it really changed how they think about when and why toxins are produced. And so you could imagine that having an impact on how we forecast whether or not water is going to be safe to use for recreation. We haven't gotten to the point exactly where that's going to change it, but I think, like, of the work that I've done, that's the one that's likely to actually have a management impact or some kind of policy impact. It's still pretty esoteric, I know. Yep. The toxins, yeah, it's a pretty serious problem. And even though we know it's linked to nitrogen starvation, I think we still don't fully understand when and why it happens. So that's still an active area of research too. On the wastewater side, I studied the bacteria that remove phosphorus from water. So we've actually made some pretty significant advances in understanding how the bacteria do that and ways that you can operate the wastewater treatment plant more effectively to make them store the phosphorus inside of their cells better so that it doesn't end up in the water that goes out into the receiving streams. And so engineers are actually using some of the work that we've figured out in how they design their wastewater treatment plants. And so that, on the wastewater side, is something I'm pretty proud of. Yeah. Yeah. And it's still microbes, because they do all the work.
So following up with that a little bit, the intersection of wastewater treatment and lake quality. So I've heard about tertiary treatment of wastewater actually being treated in a natural kind of wetland situation as opposed to in a wastewater treatment plant. Mm-hmm. So there is kind of the in-between stage between the wastewater treatment plant and the lake. So can you talk about the microbiology of those treatment wetlands and how they take care of a lot of the nutrients? Or do they?
Yeah, it's really fascinating. So the things that are in the water coming out of most wastewater treatment plants that we worry about with what respect to water quality are, of course, like the particles, the solid material that might come out. The nitrate, because a lot of nitrogen, so in your wastewater, so in your pee, there's a lot of ammonia, right? Because the urea breaks down to ammonia. There's very special bacteria in the wastewater treatment plant that convert that into nitrate. Many wastewater treatment plants don't convert that nitrate into anything else, so they just release that, mostly because they're not regulated on it. Phosphorus can come through the wastewater treatment plant as well, especially if your microbes aren't working very well, using the ones that we study in our lab. So phosphorus, nitrate, and solid particles. In a wetland, the plants do a pretty good job of sort of filtering out those particles, right? So the root system and the soil will catch it just by physical straining, right? Like they just don't pass through like a colander. And the particles get stuck in there, and then bacteria have a better chance to break them down and solubilize them and turn them into something that then would turn into carbon dioxide and other things that maybe aren't as much of a concern. And then the nitrate gets reduced to form nitrogen gas, which is inert and can leave the system as a gas. And so nitrate-reducing bacteria that live in wetlands are really, really important. And the really cool thing about a wetland system is that the plants provide the food that the bacteria need. So they actually pump sugar and other kinds of compounds down through their roots and provide that to the nitrate-reducing bacteria, which then, so they're breathing the nitrate, right? So remember I said that they can breathe nitrate. So they take the sugar from the plant and they breathe nitrate, and the nitrogen gets removed. So the plants are providing that important function in the wetland. The phosphorus is trickier. So the thing about phosphorus that makes it so difficult to remove from an aquatic ecosystem is that the chemical form that it's in is it can't be a gas, right? So if you think about carbon can turn into carbon dioxide, and so you could worry about climate change, but for the most part, that's the best way to get it to leave the ecosystem, right? It leaves, if you had sugar or some other thing that would potentially, or a organic contaminant in the water, that would leave as CO2, it's gone. Nitrate leaves as nitrogen gas, it's gone. Sulfur leaves as H2S, sulfide eventually, it's gone. Phosphorus doesn't do that. It accumulates in dead things, and it might get cycled around and around, but it doesn't go into the gas phase. And so it gets stuck in the wetland or in a lake, and it just accumulates there. It can go into the plant biomass because the plants need phosphorus. So most of those wetlands eventually need to be sort of harvested, where you take out some of the plants and weed it, right, and thin it. So, and that's the main way that you get the phosphorus out. But the main reason why phosphorus is so difficult to remove from an aquatic ecosystem is because the microbes don't breathe it. They don't turn it into a gas. Whereas almost all the other elements that we worry about can be turned into a gas, and the wind takes it away. So you have to understand the chemistry, right? Like I try to tell that to my environmental engineering undergraduates who hate chemistry, and I say, if you want to understand how pollution is mediated by microbes, you got to understand that chemistry and what the limits are that are imposed by the chemistry on the biology, right? They're just, chemically, it's just not possible to turn phosphorus into a gas.
I have a question.
Mm-hmm.
So in the beginning of this conversation, you talked about how most microbes live in communities and they offer different services for each other, or however that goes. So there's that symbiotic relationship between different microbes. Have you ever seen any evidence where microbes go to blows? Like if you had E. coli and cyanobacteria together, who would win?
We do see lots of evidence of competition. So two microbes trying to eat the same thing and trying to figure out who wins based on either how fast one or the other grows. Do they do chemical warfare? Like I think a lot of microbiologists believe that antibiotics were, evolved to be chemical warfare between microbes. So a fungus or a bacteria will make a particular chemical that kills all the other bacteria around it so that they can get the food. And some of that has been shown to be relevant in natural ecosystems. But I think that that's more rare than we would like to think. I mean, some microbiologists get really excited about it because it sounds so interesting, but I don't think that happens nearly as much as the competition, where just, two organisms are hanging out in the same place and one figures out how to grow more quickly and get the resources faster, and so it wins. And my wastewater treatment projects, we study this pretty intensively because there's two different species of these bacteria that use that accumulate the phosphorus, and depending under certain conditions, one wins over the other. And we're trying to figure out what exactly are the conditions that make one win over the other, because one of them is actually slightly better at doing its job. And so if we can figure that out more, we could engineer the system to select more for that organism that is better at doing it, help it win. So when you understand those mechanisms of competition or interaction, you could start to think more about how you would manipulate the system to achieve some goal. So that's mainly why we study it.
Okay, back to treating waste treatment in wetlands.
Mm-hmm.
Since there would, the phosphorus doesn't go away, and you have to harvest, you need I'm guessing, you would need a very large area of wetlands to treat, let's say, a small city of waste?
Probably, yeah. There's certainly design specifications that people have worked out for if you're going to have 2 million gallons a day of wastewater, how much square acreage or of wetland do you need? And but that probably depends also on whether you're building it in Florida versus in Wisconsin, right? In terms of the kinds of plants that grow there, and the temperature in terms of the rate at which the plants grow and things like that. So people who do research or actual design of those systems have a lot of that figured out very empirically. Like they've tried it, see if it works, adjust it a little bit, adjust the design a little bit, make sure it works. They don't really understand all the fine scale workings of it, but they understand it well enough to make it work pretty well. But it's true that it uses a lot of space that not every municipality has access to.
Do we have any more questions this evening? A couple up here. Could we get a raise of hand of how many people still have questions? If we should take a quick break or just plow through? How do you feel?
Doing fine.
Okay.
Have you worked on any concentrated animal feeding facilities?
I have not myself. The closest I've gotten is aquaculture, so it depends on if you call that, which I guess you probably could, right? Yeah, I mean, I could talk about aquaculture because it can kind of launch into talking about pig farms and cow farms or cow facilities. But in aquaculture, you obviously keep fish in a confined space and feed them like you would in an aquarium, like a food source that's derived from other kind of usually fish material, right? I think it's often probably harvested from some fish in the ocean or somewhere far away that is sort of grungy fish stuff that nobody wants, and they grind it up and they turn it into fish food for the fish farms here in Wisconsin, right? And so we were interested in the effect of using antibiotics to control bacterial infections in the fish, and whether or not that would influence the kinds of bacteria that you find in the aquaculture facility, right? So the hypothesis was that if the fish farm is using antibiotics, you will see more antibiotic-resistant bacteria, and you will see different kinds of bacteria where the antibiotics are being used. And what we found was that there was very little difference between the farms that were using antibiotic enhanced food, because they actually put the antibiotic in the food. They use usually tetracycline. They put that in the food. And we didn't see a lot of difference between the fish farms that were using the antibiotics and that weren't, which surprised us. And then what we did was we went and we looked at the food that was supposedly, that did not have antibiotics, and what we found that it had tons of antibiotics in it, that wasn't supposed to be there. And I think that probably is that the food source is coming from somewhere in like Thailand where they use tons of antibiotics to grow fish, and then they mash it up and send it to Wisconsin to grow. It's just kind of, in itself, kind of a scary thing. The other thing that we found was that there was a lot of antibiotic-resistant bacteria in the sediments of the fish ponds, sort of irrespective of the food source, and we did some literature searching to figure out if this was something really new, and other people had actually discovered this, and the idea there is that the conditions where it's kind of like with any confined animal feeding operation where you've got a lot of poop in one place, and it creates this really mucky, nasty condition, right? And that anaerobic, so very organic matter-rich, decomposing situation was actually selecting for antibiotic-resistant bacteria, even if there were no antibiotics there. Which was kind of a head-scratcher for us as well. Now you translate that into a pig farm or a cow farm, and whether or not antibiotic-resistant bacteria are a problem in those habitats. I think a lot of people are doing really interesting work on that, and what they tend to find is that there's tremendous numbers of antibiotic-resistant bacteria. Some of it linked to antibiotic use, which is often why people go to study it in the first place, some of it not. That just like having that many bacteria there, breaking down all of that poop from the animals is itself a thing that selects for antibiotic-resistant bacteria, which is kind of freaky. But certainly coming up with innovative ways to treat the waste from those confined animal feeding operations is something that engineers work on. And so manure digesters are a great example of that, right? Even for dairy farms that aren't confined animal feeding operations, you can use an anaerobic digester, but an anaerobic digester is a really great way to turn manure into energy because you use microorganisms under a much more controlled environment. So an engineer builds a big tank to put them in, and you control the temperature, you control the gas inflow and out, and you can harvest methane, which is natural gas, and burn it to make electricity, and control the release of what comes out of it in a way so that you're not like letting it leach into the ground or overflow into a river or otherwise either distribute phosphorus, nitrogen, potentially antibiotic-resistant bacteria, and that sort of thing. So I'm a big proponent of using anaerobic digesters anytime you have large numbers of animals in a particular location.
Considering your experience with the aqua farms or the fish farms, have you seen the recent proposed legislative changes on fish farming and what effect that might have?
Last time I worked on it was 2006, so I don't keep up with it anymore. Is there something nationally or statewide?
Yes, our Senator Tiffany and the legislature is changing the
I didn't know that.
regulations on fish farms to classify them as a regular farming operation, so it'll modify their runoff.
Wow.
And how it's going to affect streams.
That's a good thing, actually, because one thing that shocked me when I did that project was the fact that they do not have to treat their water at all. It just goes right out, and because they weren't regulated at all.
Their opinion is it won't have any effect on our water quality.
Probably depends on what that receiving water looks like, right? How polluted is it already? If there's several fish farming facilities on Lake Superior, and I would argue that any amount of nutrient input to Lake Superior will have an adverse effect because it is such an oligotrophic system that you add just a little bit and it's going to change it a lot. There are other places where if there's already a lot of nitrogen and phosphorus, then they might be right that it may not change it very much. But in general, I would prefer to see some kind of treatment happening on the effluent from the aquaculture because it has a lot of ammonia in it from the fish peeing and pooping, and it has particles from the fish food that doesn't get eaten and also from the fish poop and stuff. So you can see it in the data that it's discharging low quality water. So it's probably a good thing. Oh, it's to eliminate it. Oh, I thought you meant to increase it. Oh, no. We want to have more, more regulation. No. Liberal professor from Madison. No, the thing is that they're not regulated much already. So what's there to remove? I guess
Discharge
more.
the waters and their runoff from their operation into the creeks.
They already do. So I'm kind of confused because the ones that I was looking at, they already do. So I'm kind of.
This makes it broader.
Makes it broader. Okay. Well, I'd rather see it go the other direction.
They're reclassifying that as a farm. You give them
I see, right, because then they become, but it's interesting because it's not, the whole non-point source versus point source falls apart there because they are a point source, so they really should be regulated as such. Yeah, I see. Okay. Yep. So I'd rather see it go in the other direction.
Any other questions this evening? If not, please join me in thanking Dr. Tricia McMahon.
Thank you.